Welcome to the Bharati School online classes. In this section, we will discuss digestion of food, how food has been digested. First of all, we should discuss digestion. What is digestion? So, digestion is the process through which complex insoluble form of food has been converted into simple and absorbable form. It involves two processes: first mechanical process, second chemical process. The mechanical process of digestion of food involves mastication, deglutition and peristalsis, and the chemical process of digestion of food involves digestion with the help of enzymes. First, mastication. As we know that the chewing of food is called mastication with the help of teeth. Teeth chewed the food into small pieces. The process is called mastication. The deglutition with the help of tongue. When the bolus is formed in the oral cavity or buccal cavity has been pushed into the pharynx region, and the process is called swallowing. Peristalsis is the wave-like constrictions of food of bolus through the alimentary canal by muscular contraction is called peristalsis. These are the mechanical processes which help in the digestion of food. Next, we will discuss the first part of our alimentary canal, that is a buccal cavity after ingestion of food, where digestion has been started. So first we will discuss the digestion of food in our buccal cavity. So in our buccal cavity, only the starch part of digestion of food has been take place. It is clearly noted that only the starch part of food has been digested in this region, buccal cavity region, because of this salivary gland. Salivary gland secretes the saliva, and the saliva contains the enzyme salivary amylase ptyalin, and this ptyalin is a carbohydrate digestive enzymes, and this ptyalin converts the polysaccharides into the disaccharides. About 30% of starch digestion has been completed by the ptyalin enzyme in the buccal cavity region. The food we intake contain carbohydrates, and here we can see that this is our enzyme ptyalin, and here is the long chain we see, it is a polysaccharide. And how polysaccharide is formed? Here we can see that the single unit, and these single units combines to form, that is called disaccharides, and these disaccharides combine and form a long chain, then it is called as a polysaccharides. So here it is our starch, and it is formed by a single unit, its monomer unit, that is glucose. So here glucose plus glucose forms two dimers, that is called maltose, and these are disaccharides and combines to form polysaccharides, that is our starch. In this, here we can clearly see that the starch has been digested by salivary amylase ptyalin enzyme into maltose, isomaltose and limit dextrin. As the food we ingested, after swallowing, the food pass into the esophagus, and from the esophagus the food through the movement of peristalsis has been passed into the stomach. In stomach, food stores for about 4 to 5 hours. It is mixed with the gastric juice and be converted into a acidic pastry which is known as chyme. The bowl-like structure, when the food is mixed with saliva, then it is called bolus, and when food mixed with the gastric juice and converted into a acidic pastry, then it is called chyme. Digestion in stomach. occur with the help of gastric juice. In gastric juice, we know that the enzymes are present, that is pepsinogen enzyme, prorenin enzyme. These are the cheap protein digesting enzymes present, which is secreted by the gastric gland. The gastric gland secretes the HCl, and this HCl first of all activate the enzyme pepsin. Pepsin secrete in inactive form, that is pepsinogen. All the protein digestive enzymes has been secreted in the lumen of stomach in inactive form, because if these are released in active form, they can digest the inner lining of the stomach, and they will carry out the autodigestion. So to prevent the autodigestion, the pepsin enzyme or protein enzymes has been secreted into inactive forms. The word nogen, pro, it will denote the inactive form of enzymes. First of all, the HCl secreted in the stomach has been activate the enzyme pepsin. The pepsinogen has been converted by pepsin active form HCl with the help of HCl, and the protein present in our food, then pepsin enzyme digest it into small parts, that is peptones. But in case of infants, the digestion of proteins occur in different way, such as the enzyme prorenin enzyme has been active by the HCl renin. The pro word inactive, and when HCl present in stomach, these HCl activate the prorenin enzyme into renin form. As we know, the infants basically feed on milk. The milk contain a protein, that is casein protein. The casein protein has been digested by renin enzyme into paracasein. So the casein protein of milk has been converted into paracasein by renin enzyme, and this paracasein, in the presence of calcium ion, combines with it and form calcium paracaseinate, that is our curd. The process is also called curdling of milk. This will occur in the stomach of infants, and this calcium paracaseinate is been easily to digested by the infants pepsin, and pepsin has been, this calcium paracaseinate has been digested by the pepsin enzyme into small peptones. The enzyme gastric lipase hydrolyzes the small amount of lipids, because gastric lipase is work best in the basic conditions, alkaline conditions, but in our stomach, due to the HCl, the medium is acidic, so it is not carried as much functional. So a little bit digestion of lipids has been carried out by the gastric lipases. So we can clearly see this, the digestion of proteins basically mainly occur in the stomach region. Sorry. Next, digestion of food in small intestine. So here the main digestion of food has been take place. Chyme enters into the small intestine, and chyme enters into the small intestine and mix with the succus entericus, that is our intestinal juice, and pancreatic juice and bile juice. All the three juices are mixed in the small intestine region, and pancreatic juice and bile juice reach to the duodenum through the hepatopancreatic duct, a common duct for bile and pancreatic juice secretion. Digestion of food with the help of bile. Here we can see that what is the action of bile, how bile juice, which is secreted by liver, carried out the digestion of food. Basically, bile secreted by the liver does not contain any enzyme, yet it helps in the digestion of fats or lipids, as it carried out a process of emulsification. Emulsification is the process of conversion of large fat globules into small fat globules, which is also known as myceles. So here we can see this is our large fat molecule has been converted into small lipid droplets. These droplets are also called myceles, and the process is called emulsification. This process has been carried out by the bile salts, and the enzymes work on these fat droplets easily. That is our lipases. These lipases digest these lipid droplets very easily as the surface area is increased. As next, digestion of action of pancreatic juice. And next we will discuss the action of pancreatic juice. Amylopsin, that is pancreatic amylase, the enzyme that help in the digestion of carbohydrates. It will hydrolyzes the remaining starch into disaccharides. Here we can clearly see that the the starch has been converted into maltose, isomaltose, remaining starch, and 30% we have been studied that the starch has been converted into disaccharides by ptyalin enzyme, and the remaining starch has been converted into disaccharide by amylopsin, that is the pancreatic amylase. Sorry. Next, enterokinin enzyme that is present in the Fucus entericus intestinal juice activate the enzymes trypsinogen and trypsin. This trypsinogen in active form and trypsin is its active form. So first of all, enterokinin activate the protein digestive enzyme trypsinogen, which is present in our pancreatic juice, and trypsin activate further chymotrypsinogen and procarboxypeptidases. Here we can see that trypsinogen is inactive enzyme activated by the enterokinin. Enterokinin is secreted by intestinal juice, present in intestinal juice. So trypsinogen converted into trypsin active form by enterokinin, and chymotrypsinogen has been converted into its active form chymotrypsin by trypsin enzyme, and procarboxypeptidase converted into carboxypeptidase by trypsin enzyme. So here all the three enzymes comes into its active forms, and the proteins and peptones and proteases has been digested into its dipeptide forms by these three enzymes: trypsin, chymotrypsin, and carboxypeptidase. These are the enzymes which take part in the protein digestion, and the proteins, complex proteins, has been digested into simple proteins, that is dipeptides. And the pancreatic lipase enzyme has been converted into the fat into diglycerides, and diglycerides has been converted into the monoglycerides. Nucleic acids has been converted into nucleotides by nucleases enzyme present in the pancreatic juice. Action of intestinal juice. The enzymes of intestinal juice act on the product of the above reactions. जो इससे पहले हमने जो रिएक्शंस देखी थी पैनक्रियाटिक जूस जो कैरिड आउट करता है, उनके जो प्रोडक्ट्स होते हैं उनके ऊपर जो इंटेस्टाइनल जूस है वो वर्क करता है. यानी फाइनल डाइजेशन है वो कहाँ पे होता है? इंटेस्टाइनल जूस की हेल्प से. जैसे प्रोटीन्स हैं वो डाइपेप्टाइड्स की फॉर्म में आ गई हैं, तो उनका जो एंजाइम है डाइपेप्टाइडेज जो प्रेजेंट है इंटेस्टाइनल जूस के अंदर, वो डाइपेप्टाइडेज क्या करता है? डाइपेप्टाइड्स को कन्वर्ट कर देता है सबसे सिंपल फॉर्म ऑफ प्रोटीन्स की अमाइनो एसिड्स के अंदर. अमाइनो एसिड्स, सिंगल अमाइनो एसिड्स, दो अमाइनो एसिड्स मिलके क्या बनाएंगे? डाइपेप्टाइड्स. इस तरह से डाइपेप्टाइड्स आपस में ज्वाइन हो जाते हैं और लॉन्ग चेन फॉर्म करते हैं और प्रोटीन्स के फॉर्मेशन हो जाते हैं. माल्टोज 2 यूनिट्स ऑफ ग्लूकोज से बना हुआ होता है. माल्टेज एंजाइम जो प्रेजेंट है इंटेस्टाइनल जूस के अंदर कन्वर्ट कर देता है 2 यूनिट ऑफ ग्लूकोज के अंदर. लैक्टोज जो हम बात करें मिल्क शुगर जो होती है, लैक्टेज एंजाइम इसको कन्वर्ट कर देता है ग्लूकोज प्लस ग्लैक्टोज के अंदर. सुक्रोज जो शुगर केन शुगर है, सुक्रेज एंजाइम जो प्रेजेंट है इंटेस्टाइनल जूस के अंदर, इसको कन्वर्ट कर देता है ग्लूकोज प्लस फ्रुक्टोज के अंदर. These are the maltose, lactose, sucrose. These are the disaccharides, and these are the monomers of these simple form of foods or carbohydrates. Nucleotides are nucleotidase enzyme present in the intestinal juice convert the nucleosides, and nucleosides enzyme nucleosidase convert the sugar plus bases. Diglycerides or monoglycerides has been converted by the intestinal lipase into fatty acids and glycerol, that is the simple form of. Fully digested or semi-alkaline food is called chyle, and or we can see that the food when mixed with the intestinal juice, then it is also called as chyle. The digestive activity like gastric and intestinal secretions are controlled by the neural and hormonal mechanisms, and the sight of smell and presence of these in the buccal cavity stimulate the secretion of enzymes, and the gastric and intestinal mucosa secretes digestive hormones, that is gastrin, secretin, cholecytokinin, villikinin, etc. In this way, the regulation of hormones has been take place. The last part of this section are the disorders of and absorption of food will take place. We will discuss this in our next videos.